Hi, welcome back everyone. With everything that we have done, we are sort of primed to start our discussion of how evolutionary change can take place in populations. And what we'll do in this video is sort of look at prokaryotic populations and understand that how diversity is actually brought about into these systems. So let's imagine that we have an isogenic population. This is a word that will keep occurring throughout the course. This is an isogenic population. Isogenic means that every individual in the population has the exact same DNA sequence. So let's consider a population of bacteria. These can be very, very large, as we'll see in this video. We'll sort of try and get a sense of what are the typical numbers of a bacterial population. But let's say we have bacteria 1, 2, 3, I, J, N. So this is a population of size n. Size of a population, as we will also see, is an extremely important evolutionary variable. So in an isogenic population, let's imagine that we take individual number I, individual number J, and we have their these are bacteria. So let's say this is I, this is J. Let's also represent their DNA sequences, and so these are the two individuals, and these are population of size n of, let's say, E. coli bacteria. Now one thing is that because this is same species, same strain of the E. coli bacteria, the genome length is the same in both. The genome length is L, and if we try and match, if we begin and match the nucleotides of these two sequences, so let's say we match that there is a here, we find that there is an A here, the next to it is T, next to it here also is T, and so on and so forth. If we keep on going and these two sequences match exactly with each other as we go around the sequence, then these two individuals are said to be isogenic. On the other hand, let's imagine that in this genome length L, these two sequences match at L minus 1 positions, but there was this one position where there was a G here, but in this particular organism, this particular individual, there was a T here. Hence this particular position, the nucleotide did not match, whereas it matched everywhere at the remaining L minus 1 positions. In that case, these two individuals are not isogenic, and mathematically we define these two individuals as something that is called the Hamming distance. So Hamming distance is defined between two sequences. Both these sequences have to be of the same length. If they are of different sequences, then Hamming distance is not defined of the same length, in this case L. So Hij is the Hamming distance between sequence I and J, between sequence I and sequence J. So, in the first case that we discussed, where the sequence was exactly the same at every one of these L positions, the Hamming distance was 0 if two sequences are exactly identical. And in the second scenario that we discussed, where the two positions were identical at L minus 1 positions, but they differed from each other at this one position, then Hamming distance is equal to 1. So, when we say that we have an isogenic population, what we mean is that if we take any two individuals in this population out of these n, Hij between those individuals is 0. Their DNA is exactly the same. For instance, if I have a DNA sequence ATGCCC, this is from individual I, and if I take individual J, its sequence is ATGCCC, and then obviously Hamming distance between them is just 0. However, if I take individual K, its sequence is ATCCCC, then Hamming distance of JK is equal to 1, because there is this one position out of 6 where the nucleotide does not match. And obviously, Hamming distance between I am sorry, this should not be Ij, this should be Jk. Hamming distance between Jk. And obviously, if we were to do the same exercise for I and K, Hamming distance Ik would again be 1, because there is this one position in the sequence where the nucleotides are different from each other. So, isogenic describes a scenario where Hamming distance between any two individuals is equal to 0. So, now let us look at this following experiment. Let us imagine that we have a flask, and in this flask I add 100 mils of media, of liquid media. The idea is that I am preparing this liquid media so that growth of bacteria, I can do some experiment associated with growth of bacteria in this flask, and for that reason this liquid media has to have all the ingredients which will facilitate growth of E. coli under these given conditions. So, typically this would contain sources of carbon, nitrogen, phosphorus, sulfur, oxygen. We will have some trace elements, we will have some amino acids, some vitamins, and so on and so forth. Water obviously, hydrogen, and so on and so forth. So, and there are different ways of making these liquid media recipes to facilitate bacterial growth, but let us imagine I have 100 ml of that. To this flask I add one bacteria. Now, experimentally this does not really make sense, it's really hard to start a culture by just putting in one bacteria. As when we study experiments associated with microbiology and evolution, we will see that we typically add millions of bacteria to start a culture. But for the purpose of this discussion, let us imagine that we seeded this culture with only a single bacteria, and we were somehow able to do that. Now, that is not enough, we need to give it 37 degrees Celsius, that is the optimal temperature of growth for E. coli. We will provide it some sort of shaking so that this culture is maintained in oxygenated conditions. This just allows for better growth, and we will cover this to reduce the chances of any environmental contaminant going into our culture and competing with the organism that we want to study. That is another aspect of microbiology research, that you have to be careful of any contaminants coming into the culture, because you really want to understand your organism under those precise conditions, and obviously you have to make sterile conditions, maintain sterile conditions for that. So, under these conditions, if the nutrient conditions are just right, temperature is right, oxygenated conditions support growth vigorously, what is going to happen is that as this experiment proceeds, the bacteria is going to divide. It is going to divide first, and then we will have 2 progenies, and then these 2 will divide, and then we will have 4, and this process continues. And if we plot, and let us say we started this experiment at 5 p.m. and then after seeding the culture with this one bacteria, we went home, and we are going to come tomorrow morning at 9 a.m. Obviously, the culture is going to be just full of these bacterial cells in the flask. And in an experimental context, it is very easy to see, because the liquid that we use for growth media is transparent, it is clear liquid, and when it is full of bacterial cells, it is going to be very murky, and it will be opaque, can't be seen through. So, let us say this is 5 p.m. when we seeded the culture, this is 9 a.m. when we come the next morning, and this is number of cells. So, take a few seconds and try and sketch what does this graph, according to your intuition of how growth would take place, what does this, what would this graph look like, and just take a few seconds to sort of go through this and try and sketch it for yourself before we continue this discussion. Okay. So, what would actually happen in a scenario like this is something like this would happen, and this is referred to as 3 phases of bacterial growth. The first one, where there is hardly any growth, is referred to as the lag phase. This one, the first, is called lag phase. In the lag phase, there is no actual, or there is very little actual cell division taking place. So, imagine that the carbon source in this environment is lactose. Now, this bacteria that I introduced does not have LAC-Y and LAC-Z protein molecules to be able to import lactose and then break it down into glucose and galactose. Hence, the first thing that the bacterium needs to do is make these proteins, make them in sufficient quantity, import that lactose, start breaking it down, start using it for energy and growth purposes, accumulate enough metabolites, make enough proteins, grow in size, and only then would this bacteria divide into 2. And all of this is going to take some time. So, this lag phase is associated with the time that is needed for the bacteria to acclimatize to the new environment that it has been asked to grow in, and make the necessary cellular machinery which would facilitate that growth, and then you would begin to see growth. So, that is the lag phase associated with growth. The second phase is called the log phase or the exponential phase. In this phase, the bacteria have accumulated the necessary protein molecules required for growth in that environment, and growth is taking place exponentially, because you started with 1, this one got ready, and now this is ready to divide, it divided, you have 2, these will give 4, 4 will give 8, 8 will give 16. So, if we plot this against time, it is going to be an exponential increase, and that is what this phase of the graph captures, that this is an exponentially increasing number of individuals in the population. So, this is exponential increase in number of individuals. However, what happens is that this exponential growth cannot be sustained forever, because this is an environment in which we are doing this experiment. This has finite resources. With these finite resources, when you hit the middle of the exponential phase, what is going to happen is that there are far too many cells now. They are all competing for the remaining resources. Resources are decaying because as number of individuals are increasing, the carbon biomass is increasing, and all the carbon and other nutrients have been taken from the media to make these organisms. As a result of which, the carbon available for the individuals present in the culture now, that number is growing, is increasingly small, hence the growth rate begins to slow down. So, there is this slowdown phase at the end of exponential phase, and eventually you reach this stage which is the third phase, which is the stationary phase. And stationary phase is something that is a bit of an enigma. It's not as if that the overall number remains constant, but this is because number of births that are taking place are matched by number of deaths that are taking place in the culture media. And as a result, this number stays the constant, but there are really no real nutrients left in the flask anymore. So, this is how these numbers would grow and eventually reach something called, let us call it N final or NF. Now, 100 mils 100 mils of culture media in this flask cannot hold any more individuals than NF, and for that reason NF is called the carrying capacity of an environment. Typically in literature, the variable that you would see used for carrying capacity is K. So, this is K or NF as we described it, which just means that in these environmental conditions, with this particular species of E. coli, if you were to grow this at 37 degrees Celsius, it cannot support more than K number of individuals, and that is what the population moves towards. So, in this growth curve that we have, let us ask a couple of questions. We started with number of individuals as 1. At some point, that number reached N naught. We want to answer 2 questions associated with that. The 2 questions are: 1, how many cell divisions took place? How many cell divisions took place for the population to transition from population size n equal to 1 to n equal to n naught? That is the first question. So, I'll give you again a few seconds to think about it, and I'll tell you the answer, but let's take 15 seconds. Think about how many cell divisions took place for this transition to take place. Okay. So, I hope some of you got this, but the way to think about this is that when you have a cell and it divides, it gives rise to two cells, and in that process when we have these two cells, this one is no longer there, I have these two cells. So, what happens that pre-division, if I am looking at just this event, pre-division, the number of individuals in the population was 1, which was this cell. Post division, the number of individuals in the population was 2. This is one cell division event. What you should realize at this point is that one cell division increases the population size by exactly 1. Increases the population size by exactly 1. As a result of that, population increased by 1. So, for the population to go from n equal to 1 to n equal to n naught, how many divisions need to take place? Every cell division would increase the population size by 1. So, one cell division would increase the population, one cell division would increase the population size to 2. Then as these 2 divide, that means 2 more cell divisions take place. So, total 3 cell divisions. After 3 cell divisions, the population size would go to 4. We can also think of it like this, that we started with this one individual, we have these 2 individuals after 1 cell division. Then this one would divide, that is 2 cell divisions, and the population size is 3, this and this. But then this would also divide. So, now the population size is 3, but 3 cell divisions have taken place. This cell divided first, and then the 2 progeny divided, leading to 3 cell divisions and population size of 4. And now this will divide. So, that is the 4th cell division, leading to population size of 5, and so on and so forth. The point you should see, the pattern that you should see is that one cell division adds only one cell. Hence, to go from n equal to 1 to n equal to n naught, I am increasing the population size by n naught minus 1. And since every cell division increases the population size by only 1, I need to have n naught minus 1 number of cell divisions for this transition to take place. And I hope that makes sense to everybody. The second question that we want to ask associated with this is that as this change happened, just to go back to the previous slide. So, we can plug some numbers, that means if to go from 1 cell to 100 cells, we needed 99 cell divisions. To go from 1 cell to 1000 cells, we needed 999 cell divisions, and so on and so forth, n naught minus 1, and that is all we are doing here. To answer the second question, the second question is as follows, that as the population transitions from n equal to 1 to n equal to n naught, as this transition takes place, how many generations passed? How many generations did it take for this change to take place? And to answer this question, we should look at it in the following way, that we start with one individual, and let us call this generation 0 at t equal to 0. After one generation, this individual would divide into and the population size at generation 1. So, let us say this is the number of generations and this is population size. Let me rewrite this. So, in this representation of the system, the variables we are tracking is what is the generation number and what is the population size. These are the two variables we are tracking as the system moves forward. At this point, generation number was 0 at t equal to 0 and population size was 1 because this was the only individual we had. After one generation, we get 1 as the number of generations and 2 as the population size. After one more generation, both these individuals would have divided and the population size would be 4. After 2 generations, the population size is 4. And let us just do one more and then we will try and see what is the pattern that is emerging here. So, after the third generation, we get 4, 5, 6, 7, 8. After 3 generations, we get 8 individuals. And as you can see, what is happening is that with every generation, the number of populations or the number of individuals in the population is simply doubling. So, if we started with one individual, so in generation 0, we had one individual. In generation 1, we will have 2 times that individual, 2 times the population in generation 0, because with every generation number is doubling. So, we get 2 into what was the size of the population in generation 0? That was 1. 2 into 1, that is 2. At generation 2, our size of the population will be 2 times number of individuals in population 1, in generation, not population 1, in generation 1, and in generation 1 number of individuals was 2. So, this is 2 and we get 2 into 2, which is 2 square or 4. Similarly, at generation 3, the number of individuals will be 2 into number of individuals in generation 2. This number was 4, or let us say 2 to the power 2. So, 2 into 2 to the power 2 is 2 into 2 to the power 3 or simply 8. And we can keep on building this logic until we realize that after k generations, the number of individuals in the population will be 2 to the power k. And we can verify this by checking here that after one generation, number of individuals were 2 to the power 1. After 2 generations, it was 2 to the power 2. After 3, it was 2 to the power 3. And after k, it will be 2 to the power k. So, that is what we understand from here. So, how do we answer the question that we started with? That starting population was 1. After k generations, the population size was 2 to the power k, and I want to find out how many generations did it take for population size to become n naught, which means I want a value of k such that 2 to the power k is equal to n naught. And that is the equation that I have to solve for the value of k. And if we just do this, we can take log of both sides, log to the base 2, we get k equal to ln of n naught base 2, and that is it. So, suppose we were asking that how many generations does it take to go from 1 to 100? The answer is simply ln base 2 of 100, which if you work out is 6.67. Another way to think about this is, after one generation, this one individual will divide and the population size will be 2. So, that is one generation. After another generation, the population size will be 4. After 3, it will be 8. After 4, these 8 divide, so it will be 16. After 5, these 16 divide, so it will be 32. After 6, it will be these 32 divide, so it will be 64. And after 7 generations, it will be these 64 dividing to 128. But my question was that how long does it take for the population to reach a size 100? And now if we trace these generations, this is generation 1, after 2 generations it was 4, after 3 it was 8, after 4 it was 16, after 5 32, after 6 64. So, clearly 6 generations is not sufficient for this population size to reach 100, but 7 generations is too much, because after 7 generations the population size reaches 128, and our question is only concerned with the population size reaching 100. That means the answer has to be between 6 and 7, and that we can, so this answer 6.67, we can arrive at that by just doing this sort of a simple calculation and realizing that the answer lies somewhere between 6 to 7 generations is the time it will take for the population to reach size 100, and so on and so forth. We can, I leave it to you as an exercise, but we can check that how long will it take for this population to reach 1000 individuals, and that number of generations should be very close to 10. We will continue this discussion and we will try and get an estimate of what is the diversity of genotypes that accumulates while these generations and cell divisions are going on when we continue this in the next video.